Jared is an assistant professor in biological and biomedical sciences in the School of Science and Engineering. He works on plant viruses, and so he asked for money to go to the annual meeting of the American Society for Plant Virology. I should be able to speak pronounce that better. A top venue for plant virology, and we'll hope that had a success.
I think so. All right, well, thank you everyone, first and foremost for this invitation to be here today, and also thank you for coming. Like was mentioned, I used the funds to go to the American Society for Virology annual meeting. I actually serve on the program committee for this conference. It's a 3-year term, but that actually doesn't cover any travel costs. So this was very helpful for me to go. 2 PhD students in my lab gave research presentations at this conference. And it's really one of the best venues in the United States for virology. There's usually about 2,000 to 3,000 virologists in attendance. And this is a really great opportunity, not only for myself to try to network and establish connections that could maybe lead to collaborations or honestly improve my chances for funding, but also the PhD students in my lab really benefit from this. One student established some networking that led to a postdoc. They just graduated in December, and they started at Texas A&M University as a postdoc. So it was very beneficial for everyone who went. Specifically for me, I established collaborations with 2 investigators from Max Planck Institute. This has been very fruitful. They've shared transgenic plants with me that would take years to make. And we've been using them in our research here in recent months. And it's really generating a lot of very, I think, will be high impact data. So yeah, again, this conference was a great time for everybody, and we typically try to go to this every year. So this, I think, the conference has really led to a lot of my success. I think one of the most important things we can do at these conferences is get our work out there and establish a track record within the field that the work coming from my lab is of high quality. And since the conference, I've been awarded almost $2.5 million over two NIH grants. So this will support the lab for at least the next 5 years. And it's also, I think, attending these conferences has helped get on NIH study sections, which has been one of the most helpful things for me becoming a better grant writer. I've been appointed to the editorial board for molecular plant pathology as well. So I'm going to spend the rest of the time just giving a really brief overview of what I work on. I'm a plant virologist. I'm not doing a lot of the experiments these days. Here's the team we have. I have 2 PhD students. A third one just graduated in December, a master's student, a research technician, and I have a postdoc starting in a couple weeks who I'm very excited about. I think he's a very talented individual. So my lab has really focused on plant viruses and how they use liquid-liquid phase separation as maybe helping their replication cycle, or sometimes the host can use it against them. So this concept, you can think of this as like you shake a salad dressing. You can see these droplets forming within there. That's phase separation. So you can, in a cell, we have proteins and RNAs are just kind of evenly floating around in a diffuse, what we call a single phase. But if phase separation occurs, these can condense. They can concentrate biomolecules like RNA or protein 10, even up to 1,000-fold. And this has been of immense interest in research in the last probably 10 years, because what we're finding out is a lot of our proteins in our own bodies form nice liquid droplets, well, like they should, but you can get pathological aggregation, and this leads to Alzheimer's, ALS, Huntington's disease, Parkinson's. A lot of neurodegenerative disorders arise from improper liquid-liquid phase separation. So a lot of people have started studying this in the last 10 years, and I wanted to start focusing on this as a virologist because I think viruses a lot of the virus proteins also form these same types of aggregates. And I was curious, how does this impact replication? So in plants, this has lagged far behind, and that's often the case. We study all these things in humans, plants kind of fall up behind. So it's even been more recent. So really not until about 2020 has this ramped up. That's really when I started here at UMKC, and I've been trying to add how virus-host interactions are shaped by these condensates within cells. Most research to date really focuses on development. So here's an outline of a cell. Within a single cell, you have a ton of different condensates. Some are inside the nucleus, some are inside the cytoplasm, and these are necessary for our cells to function. When a virus gets into this cell, it has to interact with all of these, and some of them it really doesn't want to. So think of stress granules. Where are you? Right here. Stress granules suppress translation of the virus. A lot of viruses will go to great lengths to prevent the formation of these stress granules through phase separation. A lot of viruses also form their own condensates. A lot of research has been done with SARS-CoV-2, where different proteins from that virus will form condensates, and these usually have a positive role in replicating the RNA of the genome. So some of the first work I did here at UMKC was there was a protein I had studied for a while as a postdoc. It was called P26. It's from this plant virus called P-Nation mosaic virus 2. It goes inside the nucleus and interacts with a protein called fibrillarin. It forms these droplets. And it turns out these droplets are necessary for the plant virus to move systemically throughout the plant. So it has to go through plasmodesmata that connect plant cells, and that's how it goes from cell to cell, and eventually will get into the phloem of the plant to allow it to travel up to the new leaves. So this is one of the first things we did. More recently, this was just published about a month ago. We've demonstrated that these same condensates can actually shut down translation within the cell. So I'll just give a brief, sorry, this is maybe a little technical. So the big question here was we were curious, what do these droplets inside of a cell, what are they doing? They're formed by this virus. What are they doing? So we were able to actually purify them from cells. Here they are on just a microscope slide. They're GFP tagged, green fluorescent protein, so we can actually see them visually. Then we use mass spec to identify all the proteins that were in them. And when we look at the list, I don't expect anyone to know what these are. These are all ribosomal proteins. So it's basically taking all the proteins that are necessary for translation, it's sequestering them in these droplets. So I hypothesized that these should be suppressing translation. We confirm this in plants. We can actually look, the ribosome is responsible for translation in the cell. We can actually look at ribosome formation using something called polysome profiling. So here in green, this is our control. You get a nice peak. And this peak means that ribosomes are forming and they're translating RNAs into proteins. But when we express those condensates, P26 here in orange, we now lose that peak and we're losing translation along with it. So here's this is a nice image. I wish I showed it earlier. This is a single cell that we've outlined inside a plant. Here's the nucleus. And these green dots are the condensates formed by the virus. And they are huge. They're the size of the entire nucleus. And there's usually about 2 to 3 per cell. So what we've come up with is this model where these things are basically like a sponge sitting in the middle of the cell, pulling everything into it, and then disrupting translation in the process. So we've used the mass spec to identify all the proteins and RNAs. We use sequencing for the RNAs to figure out that translation is being disrupted. So if you think about viruses, we were trying to think, well, why would the virus want to shut translation down? And I think that's because it doesn't really, translation of a virus RNA and establishing a systemic infection are incompatible. If a ribosome is translating the RNA, it cannot move through the plasmodesmata and establish a systemic infection. So early in the infection, you want RNA replication, you want translation. During late infection, you want to now start forming virus particles that then move and establish a systemic infection. And because phase separation is concentration dependent, so we identified a concentration, it's about 12 micromolar, that is required for droplets to form. Anything less than that, you don't have droplets. So you only reach this concentration late in the infection. So now the condensates form, and now it's going to favor virus assembly and systemic movement instead of translation. So that's one way that viruses will use condensates to kind of promote replication. Another study that we've published was a protein from the cell that can form condensates and then inhibit a virus called turnip crinkle virus. And this was it's a very descriptive name. It doesn't actually just infect turnips. A lot of plant viruses are kind of promiscuous. They'll infect all sorts of plants, but the symptoms are just that. It will crinkle the leaves. So these are really simple viruses that I work with. If you think of coronavirus, it's about 28,000 bases long, encodes probably at least 30 genes. Turnip crinkle virus is 4,000 bases long and encodes 5 genes total. But I like studying these because if you only have 5 genes, and for reference, humans, we have about 23,000. If you only have 5 genes and you have to get everything done, to replicate this virus, most of these proteins are multifunctional. They're the jack of many trades. So that's kind of why I've enjoyed studying some of these model plant viruses. But we screened, go through this, we screened a bunch of proteins for antiviral activity. And one that we focused on was called Cas3. It's part of the splicing machinery that will splice RNAs together after they're transcribed. We found that it could inhibit our virus at least tenfold when we overexpress it in plants. We could also knock it down or knock it out of the plant, and the virus would replicate better. So we could kind of get it both ways. And it formed really nice condensates again inside the cell. Blue here is the nucleus. This is a GFP tagged protein. And we can see these, we can spot these on microscope slides. We can make these phase separate in vitro. When we purify the protein, we can just put it in a test tube. You can put in any crowding agent, and that will force it to form droplets. So we can measure all of this in a test tube, which is pretty nice.
The crowding agent is basically making the local concentration higher.
Yes.
Okay.
Yep. So we use polyethylene glycol for that. We could mutate this protein to make the condensates very small. This is in cells. So here's the normal size. Here's the mutant version. And when we did that, we could actually see a rescue effect that the virus could replicate a lot better in the presence of those smaller foci. So the antiviral activity here of these condensates corresponds with their size. We again used mass spec to see what proteins were found in these condensates. We found 2 pretty common proteins, heat shock protein 70 and GAPDH, that are necessary for virus replication. So our model is that the condensates are sequestering these 2 factors, not allowing them to be used by the virus. We confirmed the interaction. This is called biomolecular fluorescence complementation, or BIFC, where we put half of fluorescent protein on Cas3, half on HSP70, and if they bind, you'll get complete fluorescence, and you can visualize it with a microscope. So it's a pretty nice way of confirming mass spec results. We proved this kind of with when we overexpressed HSP70 or GAPDH, we could get a rescue effect. So that alleviated any of the antiviral activity that we saw from Cas3. I think this is one of the nicest ways to view how antiviral Cas3 could be. So we wanted to test some other viruses. So we used tobacco mosaic virus. We engineered tobacco mosaic virus to express GFP. So we can infiltrate this in a leaf. We just inject it with a syringe to do 2 spots. On the left spot, virus by itself. On the right, virus plus Cas3. So the higher the GFP intensity, the more virus replication that you had. So we can see this with just a UV lamp. We don't have to do fancy things like real-time qPCR or anything like that. So it turns out Cas3 is broadly antiviral. It's not that it just inhibits turnip crinkle virus. It really could inhibit any plant virus that we tested. So here's the summary of the second part. These condensates that form in the cell by Cas3, they can sequester GAPDH and HSP70 or host proteins that are necessary for RNA replication. So they reduce virus accumulation about tenfold. So that's just kind of a taste of what we work on in the lab. I'd like to need to thank all the people in the lab that have worked on these projects. And again, thank you very much for the funding to support my travel to ASV. I appreciate it. Thanks.
Thank you, Jared.
Yeah.
That's interesting and apparently very rewarding. This is an amazing outcome of the injection of a little bit of funds into a project.
No, I'm very excited for the next few years. I think we have a good opportunity to make a lot of headway. Good.
You have loose funding that you could funnel back?
Kick back? Probably. We probably could. I don't know though. The team's getting bigger, so the salaries eat it up.
Just a way to ask. Thank you.
Yeah.
Any questions for this technical expose? Bonnie?
I'm just curious how what you're doing for plant research will eventually potentially impact for animal virus.
Yeah, so I think my main, because again, it is funded by NIH, there has to be some aspect of human health. So most of the pathways that I study that the virus is kind of targeting or disrupting are broadly conserved. So the one we're focusing on mostly now is called autophagy, which is in pretty much every eukaryotic system. And the thought is if we can better understand how these model RNA viruses can disrupt that pathway, we can make those same applications with SARS-CoV-2. And actually, the viruses I work with, they're in what's called the flavivirus superfamily. So they're actually closely related somewhat. So West Nile virus, Zika virus, dengue virus, those are of more medical importance.
Sully.
It sounded as if inside a droplet, it's a pure droplet in terms of one kind of protein. Do you know that's the case?
Actually, we think they're very heterogeneous, that there's a lot of, so when we do mass spec, we can typically at least see upwards of 200 different proteins that are in a single droplet. The same goes for RNAs.
Single droplets for mass spec?
Not so we could do them with FACS, fluorescence activated cell sorting. You can actually separate the particles on that. I have not done that. We have done it in bulk.
Okay.
But I assume that most of them are similar.
But that's an assumption, that not necessarily.
It's an assumption.
Sully's point may still be valid. Some of them may be pure.
It very well could be.
Okay, no more questions. Thank you.
Thank you.
Jared.